And we're going to continue that from where that just ended. So we have water in there, tube number 1 of our 11. We're at our maximum absorption of 620 nanometers. And now what we want to do is we want to kind of go through our chart. So we want to know, what is my absorption and what is my transmittance? Well, I know for water, my absorption should be absolutely 0. That's what's being absorbed. So if you think about it, if nothing is being absorbed, how much is actually being transmitted or passed through it? Well, if nothing's being absorbed, everything should be. My little toggle switch, all I'm going to do is flip it up, and it's at 99. Should it be 100? Absolutely. Are these machines not as calibrated as they should? Yeah, they're going to be close. There's always going to be a little scientific error. I would still put 99. It's not like it says 27 or something really, really far off. It's at 99. So my absorption for plain water at wavelength 620 is 0. My transmittance is 99. Then all I'm going to do is kind of go through and switch tubes. I'm going to go in. I'm going to switch my toggle switch back to absorption. I'm going in and putting tube number 2. I do not have to re-zero. I'm at my maximum wavelength now. So now my absorption as we go through is going to be 0.09. My transmittance, 80. Okay, so now we're at 80% that's actually being transmitted through. That's all we're doing for all 11 tubes. I'm going to go back to absorption. I'm taking out tube number 2. I'm going to put in tube number 3. I am not switching wavelengths here. Why? Because 620 was my maximum absorption for that particular wavelength. Now I'm at 0.15 for absorption. Transmittance, flip the little toggle switch, I'm at 71. Kind of go through, absorption back down, taking out tube 3, I'm now putting in tube 4. So now my molarity is 0.3 for this particular tube. Close it, I'm at 0.24 for absorption, which means my transmittance, how much of that wavelength is actually being passed through of that particular setting. And now we're at we're just at 5.8 is my percentage transmittance. Open that up, I'm going to switch back to absorption. I'm pulling out tube number 4 and goes tube number 5. Close it up, 0.35. My transmittance, 0.44. So you're going to notice that as the absorption goes up, the transmittance should go down. I'm going to go back to absorption. I'm going to open up my door, putting in the next tube, close it up. This is tube number 6 now, so my absorption is 0.42. My transmittance is 37%. Going to tube number 7. Let's go back to absorption. So we only need to zero it out with our first one, which was water, which was our blank. We're staying at our maximum absorption. I'm not changing wavelength. Now absorption for tube number 7 is 0.50. My transmittance is 31%. Going back to absorption, going back to my next tube. So we're now up to tube number 8. Tube 8 goes in. Tube 8 is 0.7 molarity. Our absorption is 0.59. I can pull out this sample of copper sulfate. I'm sorry, let's find our transmittance. So we'll switch that to 0.62. So for sample number 8, change your absorption to 0.62. Transmittance is 24%. Going back to absorption, opening it up, tube number 9, sliding it in. My absorption here is 0.65. My percent transmittance should keep going down. We're now at 22. We're up tube number 10. My molarity is 0.9 here. So if we change back to absorption, put that tube in. My absorption is 0.71. My transmittance is 19%. And I'm now down to my 11th tube. My 11th tube is straight copper sulfate. I'm switching that back to absorption. Copper sulfate's going in. My absorption is 0.78. My transmittance is 16%. Okay, so with this particular section now, with all 11 tubes, you guys are going to go through and analyze some data. So it says make 2 separate graphs, paste them into this section. Once again, go to Google Sheets or get some graph paper from me. Please do not use that line notebook paper and make your own scales along the way. Graph A, you're going to do absorbance versus concentration. I tell you what's on the X, what's on the Y axis. Make sure you give them a good title. Graph B, percent transmittance versus concentration. So here's my concentration. There's my absorption, there's my transmittance. So you're doing this versus this, and this versus this. With that, you guys are going to utilize it to answer some questions along the way. Okay, so it's going to ask you about the data that you find in those graphs, as well as some other things. Go back through the notes. That may help you a little bit. Make sure you guys are reading through the lab. There's vital pieces of information that may help you guys answer questions within that. Okay, any questions, you guys do know where to find me. If you need to go through and talk about this machine a little bit more, or go over parts of it, or you missed part of it, or couldn't quite see something, please, please, please let me know. Thanks.